Okay, here's round 2 of cellular respiration. So I think just to go back for a second, remember we just talked about redox reactions, which use these carrier molecules such as FAD and NAD to take electrons, to pick them up. So when they're in their oxidized form, right, like this FAD+, it means it can pick up an electron. And then when it picks up the electron, very often with some protons attached, we can find it as FADH2, and now it's in a reduced form. So that's the redox reaction quick summary. I'm going to use the slides as they are here rather than to put up a slideshow because it doesn't always show the full slide, so I want to be sure you can see that. And actually, I might get rid of me here for a second, if I can just kind of remove me. So let's see if I can do it. Oh, no, that didn't do it. All right, let's go back to this. Okay, I guess we're going to go with it, sorry. So the next part we talked about was that ATP, which by now you're pretty familiar with, is an energy molecule, and that its structure has 3 phosphate groups attached to it. So here's its structure. Here you can see a ribose sugar. That's what this is right here. And this is a nucleotide, or just a part of a molecule here that is combined with this ribose sugar. And then we have these 3 phosphates, 1, 2, and 3, alpha, beta, and gamma. And each one of these are attached to each other by a very high energy bond. So that idea is that they are not too stable. And when ATP is hydrolyzed with using water into ADP and a free phosphate, that process releases energy. It's exergonic. So remember, this would be a reaction that would have a negative delta G value. In other words, it's a catabolic reaction. And so it's exergonic, and it's going to release some energy, which then can be provide energy for the other type of reaction that needs the energy, which is an endergonic reaction, sometimes an anabolic reaction. And when the energy release from this ATP hydrolysis is coupled with an endergonic reaction, which would have a positive delta G, it helps that reaction to move forward. So there you go. So that's how the energy from ATP is used. Now, one of the things that happens a lot in biology and in the cell is either the addition of a phosphate group or the removal of a phosphate group from different molecules. So when something loses a phosphate, it is what we call dephosphorylated, or it's a process of dephosphorylation. Boy, that's a big word. When a phosphate group is added to another molecule, that's called phosphorylation. When you phosphorylate something by adding that phosphate group to it, so here we see this green blob here, this molecule of whatever it is getting phosphorylated. You can see it here is phosphorylated. What that does essentially is make this molecule less stable and therefore much more likely to react. So when we see those series of biochemical reactions where we go from A to B to C to D to eventually some product, very often this is a part of the process, is putting one of those intermediate products into a state that will now react and go to the next level, or to the next compound or intermediate product that it's going to make. So that's what phosphorylation is. Now, where things eventually, how ATP is generated is really the big topic here of cellular respiration. So ATP has to be made because it's always being used, so it has to then get regenerated. And where there's ADP, we can have the addition of a phosphate group go on to ADP to form ATP. But this is an endergonic reaction. So that's a little bit strange because we're trying to generate this molecule of energy. But in order to do it, it's going to take some energy. We're going to need to get this process is an uphill reaction to get to ATP. So it too is very often coupled with some other reaction that's going to release enough energy so that that can happen. So the energy required for this reaction, this coupled reaction, is going to come from some other reaction that will be exergonic. And it can be what we call substrate level phosphorylation. So that's one way it can happen. Or it can happen by this process called chemi. You notice the chemical part here, chemi osmosis. And you understand what osmosis is. So these are two ways that ATP can be made. Most of it, as you can see here, is formed by chemiosmosis. And where that happens is in this thing, if you remember the mitochondria, the powerhouse of the cell. A lot of this type, the substrate level, which I actually at this moment I'm going to make bold because, and italicize it, because it's really an important concept, so it shouldn't be just in brackets. This is a substrate level phosphorylation that can happen, so that's an important one. Or the other type, which is chemiosmosis. So here, let's go to the next thing. So here is cellular respiration in a nutshell. And what we have is the formula, the chemical formula, where we see one molecule of glucose combining with 6 molecules of oxygen, forming these two waste products, carbon dioxide, 6 molecules of carbon dioxide, 6 molecules of water, and then the desired product, approximately 36 molecules of ATP. So from one molecule of glucose, the cell can make about 36 ATP, which is pretty good. Now, how we get there is a series of what I kind of refer to as a 3.5 act play, where glycolysis is act 1, and that takes place in the cytoplasm or cytosol. This oxidation of pyruvate, which I sometimes will refer to as, and I'm going to do this right here so I get to edit as I speak, the link reaction, which is more often what I call it, but it's just oxidation of pyruvate. So now that you have a little understanding of oxidation, the link reaction, these are the same, and the citric acid cycle, so these two things are going to occur. And citric acid cycle is also called the Krebs cycle. So we're going to put both of those terms here so that you remember when we say Krebs, really we're talking about citric acid cycle. Obviously this is somebody's name who discovered some of this. And then finally, the last act of this play is this one that's called oxidative phosphorylation, which we will see shortly. Sometimes I call this oxphos because it's just easier. All right, let's move on. So let's start with Act 1, and that's glycolysis. And glycolysis happens, as I said, it's going to happen in the cytoplasm. So that's really important because that's where the scene takes place. And another thing that's important in glycolysis to remember here, and as you're learning, is that oxygen in this case is not necessary. So when we look at the equation for cellular respiration, we know that oxygen is involved, but it's not involved here. So in glycolysis, what we're going to see are 10 enzymatic reactions. So in other words, 10 steps, each one of them catalyzed by a particular enzyme. The short version is that we're going to put in a glucose and work together with the oxidized form of NAD, 2 of them. We need actually a little startup energy. There's the ATP. And we need this ADP as a substrate to make a little bit more ATP. So you'll see this in a minute. And then the outputs, in other words, the yield of this reaction of glycolysis is that we're going to get 2 molecules of something called pyruvate. We're going to get 2 reduced forms of NAD, so NADH. We actually generate 4 ATP, but we're going to see we already spent a couple, so we'll see the net yield here. And then we're going to have a couple of ADP at the end. So that's kind of very much the short version. Here's the dazzling first half of that, and this is the first 5 steps in which we take glucose. So we're looking at their structural formulas here. And in the very first step, that very first enzyme, which I've mentioned to at least some of you before, hexokinase. Hexo has to do with the 6 carbons of glucose here. And then a kinase is an enzyme that has to do with either phosphorylation or dephosphorylation. So this reaction, if you notice, ATP is used in the very first step, which means that very first step would be endergonic and needs ATP. This is a coupled reaction where the energy released from ATP hydrolysis, so here's the ATP going to ADP, is going to push this reaction that would not normally be favorable. So this would be the endergonic piece of it, going from glucose to glucose 6-phosphate, but it does happen because of this. So glucose becomes phosphorylated right here. So now glucose becomes this thing called glucose 6-phosphate. Then glucose gets rearranged a bit by an enzyme called an isomerase. So isomers are just different forms of the same molecule, just rearranged. So here we have fructose 6-phosphate. We didn't lose anything, we didn't gain anything, we're just rearranged. And then step 3, notice we have ATP coming in again. Here's another coupled reaction using another kinase. This is phosphofructokinase, but at this moment, don't worry about that. And now we see that phosphate that's removed from ATP is going to phosphorylate fructose 6-phosphate into now fructose 1,6-biphosphate. Then we have another enzyme that's going to split it. So it was a pretty symmetrical molecule at this point. Now it's going to get split. But we need one more enzyme to make both of these into the same thing. A little bit of a rearrangement again of one of the 2 of them using an isomerase. And then we end up with something called glyceraldehyde 3-phosphate. For short, we call it G3P. Okay, so a lot to take in there. Hopefully you can review this and that'll be helpful. And I kind of put this back in. This is where here's where the action is. Here's scene 1 is right out here. So glycolysis, so far we've only done half of it, but we have a glucose that comes in and it's getting converted here in the cytosol or cytoplasm. Okay, so just so you know, cytoplasm and cytosol are kind of used somewhat interchangeably, but really cytoplasm means everything within the cell membrane, all the stuff that's in there, include the organelles, where cytosol is really referring to the watery part of that. So this whole thing, this first 5 steps that we just described is called another text called the preparatory phase. Because we're investing some energy, ultimately, hopefully, to get more energy. But so far, all we've seen is 2 ATP being actually spent. So that's not productive if our goal is to make ATP. So this is called the preparatory phase. We're getting ready, we're getting set up, we're creating molecules that are now going to be able to keep going, and hopefully we'll be able to take electrons from them, we'll be able to oxidize and phosphorylate some things to make some ATP. So here we go. All right. So that's the preparatory phase. And then we go on and we have this payoff. And the payoff phase is the second half of glycolysis.